Welcome to Library Seminars, NOAA Central Library's webinar series for the presentation of research and ideas in support of the National Oceanic and Atmospheric Administration's mission. Today's presentation is titled Leveraging Scientific Uncertainty for Estimating Among Assessment Variation in Overfishing Limits. The presentation is part of the National Stock Assessment Science Seminar Series, which is developed by NOAA Fisheries. Today's speaker, Kristin Privitera-Johnson, will be introduced by the organizer and host of the series, Kristin Blackheart from the Office of Science and Technology. Before I hand over the mic, here are a few housekeeping items for our live audience members to improve the webinar experience. If at any point you have trouble with the audio or visual components of GoToWebinar, log out and rejoin us. This resets the software and usually resolves most technical issues. This presentation is being recorded and will be available on the NOAA Central Library YouTube channel later today. I will add the channel's link to the chat box. Our speaker shared her slides with us, so please feel free to download them from the handouts menu in the webinar control panel. And most important, we're very interested in your questions, and we encourage you to ask them throughout the seminar, even though the speaker will not address them until the end of her presentation. All audience members are muted, so type your questions or comments in the chat box under questions located in the webinar control panel. So let's get started. The mic is yours, Kristin Blackheart.
Thanks, Lisa. I'm really excited to have Kristin with us here today. For somebody who is still so early in her career, she has a lot of really cool experiences on her resume. She spent some time putting sharks on what qualifies as a treadmill. She did research on corals in the Caribbean. She's kind of been all over the place and done all sorts of cool things. And now she's doing some really cool applied research in the Punt Lab at the University of Washington. What I'm really excited about is that she's here today to share her master's thesis research with us, which hasn't been super widely shared. So it's a great opportunity for us to hear some of that applied research that she's done on scientific uncertainty in assessments and its application to overfishing limits. So with that, I will hand it over to Kristin, and thank you so much for being here today.
Thank you so much for that introduction and for the technical support, both Lisa and Kristin. Disclaimer for the audience, I'm in Southern California today visiting my folks, and it is the time of day where the leaf blowers are coming through the suburban neighborhood. I hope they're almost done, but I'll try to talk over them. Kristin or Lisa, let me know if it becomes untenable. Some things I just can't control for. And just like Kristin said, this work that I'm presenting today finished around the end of 2019, and I think we all know what happened early 2020, so I didn't quite go on that press PR conference circuit one might usually do when finishing a project during their master's or PhD. And so I want to start with the acknowledgements, just to let you know what the structure of the funding and support was for this project. I received the National Science Foundation Graduate Research Fellowship Program funding for 3 years, and that's what helped me work through my master's and get connected with Andre Pont at SAFS. Usha and Rao Varanasi also supported my last quarter to get a second chapter written and published, so thank you so much to them. The Pacific Fishery Management Council also supported my travel. Towards the end of the talk today, I'll share a bit about what the tactical version of this research looked like as I presented it and got reviews and feedback from the SSE at that council. And my master's committee consisted of Dr. Andre Pont and Timothy Essington, both professors at UW and at SAFS, and NOAA's very own Dr. Jason Cope at the Northwest Fisheries Science Center. And I've kind of broken up what I'd like to talk with you about into 5 chunks. I'll talk about what we call the P-Star Harvest Control Rule, quickly define it, and then show how it has been used as applied at the Pacific Fisheries Management Council. We'll do a little bit of an uncertainty roundup. I'll talk about some of the common flavors of uncertainty and maybe point out where the method in step 2 could be underestimating uncertainty, which will lead us right into what I did for my master's, which is a projection-based approach. And then finally, like I alluded to before, talking about what this academic research endeavor morphed or was adjusted for a tactical review for the Science and Statistical Committee at the Pacific Council in 2019. So let's get started with the harvest control rule and kind of like the big picture of so what. So the so what is one of our important objectives of the Magnuson-Stevens Act is to avoid overfishing. And so we often see this as there's an overfishing limit. That's our absolute ceiling. Here comes the leaf blower. And then there's a ABC control rule, a type of harvest control rule that helps give us some buffer between that overfishing limit to account for uncertainty in things. That's what I'll be talking about most today. But as you folks well know, that there's also the annual catch limit that can be specified, and depending on your region, your regional fisheries management council and your stock of interest, you might even have some further subdivisions for your catch limits, like a harvest guideline or an annual catch target. But like I said, today's work all operates, how do we get a buffer between the overfishing limit and our ABC? Okay, and so I want to start with a simple drawing. So some scientific uncertainty in overfishing limit estimates. That is in the definition of the OFL and the ABC, as stated by the MSA national standards, is that we have to have some scientific uncertainty kind of backing up what we're doing. And so let's just look at a distribution of catch or overfishing limits centered on this overfishing limit itself. And that distribution has some width, some standard deviation, and that's going to be the metric for scientific uncertainty that I talk about today. And the probability-based harvest control rule, or P-star version that I'm talking about today, also has a component that takes into account the manager's risk tolerance, which we usually call P-star, probability of overfishing. There's some constraints. For example, they cannot pick a probability that is bigger than 0.5. And so everything that I show you today, the shaded area under the curve, is going to be for a P-star of 0.45. And so let's make this a little bit more complicated. Scientific uncertainty is important here because it's how it plays a big role in how that ABC is set. So that red line, dotted line I've just added here is the quantile that is associated with that P-star selected by the managers, right? And so let's take a look at what happens. Here's that same plot with less stuff. What happens if there's more uncertainty? So you increase the standard deviation for this distribution. And what if you increased it even further? Let's take a look at, these are all the same 0.45 P-star. This is what you just saw. This is for that middle flattened curve and the flattest curve. As you can see, those dotted red lines are becoming smaller proportions or an increased buffer away from the overfishing limit itself. So how do folks do this? What are some ways that people typically quantify scientific uncertainty, define it? You can look at the variance of assessment model outputs given the assumed model structure. So you might see this as asymptotic statistics, bootstrapping, Bayesian methods. You can also take a look at a particular model from a set of competing models, and that we usually look at sensitivity tests. You could use model averaging, all sorts of ensemble model things, and Monte Carlo methods. But I think I want to talk about, the origin story of how this rule was applied for tactical use at the Pacific Fisheries Management Council, to keep explaining the thought process behind what I ultimately did for my master's. So let's take ourselves back to 2011. I believe this analysis was prepared in response to a pressing management need that came from a requirement from the reauthorization of the Magnuson-Stevens Act to implement these annual catch limits by 2011. So some of the folks here listed as authors on this fishbowl publication were on the SSC at the time at the Pacific Council, and they thought, okay, we would like to use this P-star rule for our stocks that we review. How should we quantify the uncertainty? And they noticed, hey, here's some biomass time series for Pacific whiting. Each line is the time series that came out of a different assessment, and I think there's 15 assessments represented on this plot. And the big takeaway is, well, historical biomass varies among assessments of this same stock, and it is true for the groundfish and coastal pelagic species that they work on at this council. And so they thought, let's quantify the variation across these different spotting biomass, historical biomass time series. And they actually tested 3 different methods for doing this and picked their second one. So the one that I'll focus on today is this. We're assuming that the mean of the biomass estimates in a year is the best estimate of the central tendency. And so to get at the variance, all we need to do is think of variance in biomass as a measure of the square deviations from the annual mean in log space. And so, there are a lot of strengths to doing this. You can define that sigma, as I'll call it, that standard deviation of that OFL distribution using historical biomass. And it's pretty useful for getting at uncertainty in the biomass itself. It's useful because it was a pretty reasonable and logical first approximation for accounting for uncertainty and preventing overfishing, which was one of the big goals of the reauthorization. And I'll expand on this blue point later, but this method incorporates a lot of the factors that lead to especially model uncertainty in assessments, especially assessments of the same stock over time. And so I'll kind of wrap this section up with, this is how the Ralston et al. Analysis was applied. So the groundfish and coastal pelagic species at the Pacific Council have used a tier system based on data availability, and they call them categories. So a category 1 stock usually has information about catch at each age, or maybe at catch at each length or both, and other relatively informative data. And so the Ralston analysis, when they took a look at a bunch of stocks and the variation in their historical biomass, determined that 0.36 was going to be their value. And they thought, well, you know what? That's when we have a lot of information. What about when we have a little bit less information? So a category 2 stock, you have some biological indicators. Oftentimes in this council, you'll see data limited stock assessments used. And the logic for defining the width of the OFL distribution here was to double that 0.36 that came out of Ralston et al. And there's a third category, and if you have a knack for number patterns, you see we double it again for the category 3 stocks where you have few available data, an example being maybe just landed biomass. So let's look at this same information in pictures. So I've adjusted the widths of these 3 potential OFL distributions to match the 0.36. So this is the narrowest one, the 0.72 and the 1.44. And we can see that the red dotted lines, which are still the ABC, are going to be different depending on which category the stock is in. So let's talk about, we talked about some of the strengths and just how this has worked in the past. Let's talk about how we could be potentially underestimating uncertainty by using this method. And I'll start with that uncertainty roundup that I promised. So we have this ecosystem, and our fisheries management process maps onto it, maybe cartoonishly like this, where we collect data on an observable part of the ecosystem that we care about, where the stocks are living, and we're a part of it too. We use that collected data to assess the stock status of the stocks that we're interested in, or need or require, that is then used to produce scientific advice to set catch limits. And then those catch limits go hand in hand with implementing regulations and creating a list of research and data needs that then feed back into collecting data again. With that defined, I'll start with the first flavor of uncertainty that you can get in a system like this, and that being the process uncertainty. It's inherent to all natural systems due to the underlying stochasticity and population dynamics, so maybe like unpredictable variation in recruitment, growth, and natural mortality. And many describe this as an irreducible and uncontrollable uncertainty. It's just a fact of life. And I usually picture process uncertainty in the ecosystem box here. There's also observation uncertainty, which I associate with the collection of data. You can think of it as the variation in measurement of observable quantities like catch or size at age. And we have a couple of things that happen in the assessed stock status phase. So something like model uncertainty, which is the misspecification of things like model parameters or even the structure of the model. An example being like, think about assuming the incorrect form of selectivity as a function of fish size. We have estimation uncertainty, which is the inaccuracy and imprecision in the estimated model parameters themselves. And then I've added these two as implementation uncertainty is definitely talked about a lot in the literature, right? That's over here where maybe there's under-attainment, over-attainment, we're not quite sure how things are going to play out on the field after the catch advice and rule setting phase. And then this communication one I think about as existing between the stock assessments and communicating those stock assessments to folks that need to make decisions. And I think there are some uncertainties there because some techniques that we use take a long time to get. Just think at the SSC level, right? They review new methodology for quite a while to make sure it's robust. And it just kind of, I think of that as kind of expanding time, to the amount of time it takes to explain that to more stakeholders in that community over time. But I'm going to focus, I think the technique I'm going to be talking about, the method here, mostly addresses these uncertainties in gray, and I'll generally refer to these as scientific uncertainty. We don't look at implementation or communication with this work. So, like that Pacific whiting plot I showed you, we saw that assessments for the same stock can vary in their spawning stock biomass, and I'll eventually show you in overfishing limits. And why does that happen? The first reason could be the chosen model structure that was used to conduct the stock assessment, right? So I grabbed this picture from the New England Mid-Atlantic Council website, and we have so many tools we can use now to assess stocks. The ones that I talk about today will definitely be the single stock species spectrum of models you could use. But I think in a couple months, my lab mate Grant Adams will come and talk in the seminar series, and he's working on multi-species things with the Alaska Fishery Science Center, Kristin Holzman and her colleagues. So, there's the new model structures that we're building to address all sorts of things like predator-prey interactions, habitat, climate change considerations, end up tweaking those model structures, over time. And so we're going to get variation in what, maybe Boccaccio rockfish, what model was used to assess it in the early 2000s versus, maybe it will get assessed again in 2023. It's going to be a different model structure. Two other things we can think about are like fixed parameter values. Here's the famous Pope derivation of the fixed value for natural mortality as tweeted by Trevor Branch, a professor at UW. We fix the parameter values that are difficult to estimate, and that value can change over time based on new information. what have you. We also might use prior distributions to select values for other parameters, and that instantly made me think of the steepness meta-analysis that folks have used for West Coast rockfishes to get a value for steepness based on what we do know about other conspecifics in the rockfish family. We can also get variation in biomass and overfishing limits from the kind of a data we have available. I stole this from the sablefish, the most recent US West Coast sablefish assessment, where the data available for the assessment scientists might change over time. New ones might join, some might stop due to whatever reasons. And we also get variation because of the type and software used. And so stock synthesis has everything that I use today, or that I described today, used stock synthesis as the software structure, but even within that, for assessments over time, we're seeing changes in the development of that software, which might, the improvement of that software that affects our estimates. And so the final two things I'll talk about as influencers of variation in these two quantities are the stock assessment team that put together the stock assessment. Now I'm cheating. This is actually a picture of me and Chantal Wetzel presenting the Sigma science that I'm talking about today at the SSC. And so we're not doing a stock assessment review here, but it's a convenient picture I had. Another group that might change over time. So stock assessment teams will change based on, personnel's availability each assessment cycle. Scientific reviewers on the SFC, for example, also cycle through their roles, and with them come the different areas of expertise, priorities, and whatnot. And so that is something that we know can influence our estimates as well. And so I want to tie in that science, where does scientific uncertainty become associated with calculating the overfishing limit itself? And so in the simplest way, the overfishing limit is defined by your exploitable biomass, the biomass you can actually access, times your fishing mortality, which in this case today we'll be talking about F proxies for MSY. And here in this cartoon, all these gray fish, think of these as maybe all the spawning biomass in the stock that we care about, but we can only access, these ones in this blue box. Think of this as selectivity and all sorts of things like that. And then when we multiply it by the fishing mortality proxy, that the fish that are inside this red-orange circle, those are the ones that we would have access to in the catch limit, right? So some caveats. So now that we've kind of looked at two important quantities and subsets within that define the OFL itself, we can look at some of the caveats to using the historical biomass approach that Ralston put out. So it uses historical biomass versus exploitable biomass. There is a subtle difference there, which I'll talk about a little bit more. It also doesn't use the OFL itself. Instead, we're using the historical biomass as a proxy for the uncertainty in the OFL. It also only includes the uncertainty associated with the terminal year biomass, which has been shown to lead to negatively biased estimates of that sigma for the OFL distribution. It also assumes the estimates of our FMSY proxies have negligible error themselves, and all of the uncertainty is coming from uncertainty in biomass estimates alone. Okay, and so what can projections do? So I've told you caveats about using just historical biomass. What I'm asserting today is that we can use projections to address more uncertainty and get at some of the caveats I just outlined. So we can directly project the exploitable biomass itself. We can apply the assumed FMSY proxy, though I would probably earmark that there are probably areas to improve upon the projection-based method for this assumption that there's negligible error in FMSY proxies. We can use things like F30%, F45%, F50% in this method, so we can apply the proxy directly. We can also capture uncertainty in the current abundance and age structure. So here's a cartoon model of what age structure could look like, where we have different age bins for fish and the recruitment that's generated from the ages that can produce, surviving between age classes. I think one of the last seminars was led by Lee Cronenfine, who works out a lot on size structured models. So you could also have a model like this with size transition matrices going between different sizes of your critter of choice. And another advantage to the projections is not only can you look at uncertainty in these structures themselves, but then how those structures change over time. Whether I'll be using age structure in this method. All right, I've alluded to it a lot. Let's get into it. What is the projection-based approach for quantifying uncertainty? And I'll present what I'll call the academic research questions first. These are the questions, or the way that I present how these questions were addressed will be from the academic exercise, and then I'll talk about how they were modified to be actually more tactical and usable at the council level. So what we cared about were how do projections of spawning biomass and OFL, how do they vary among assessments of the same stock, among assessments across multiple ground fish stocks? What if we started the projections from different years? How does it vary based on having different start years? And then, you know what, we can have deterministic and stochastic recruitment using the RecDevs from stock synthesis report files. So why don't we take a look at the effects of that as well? And then ultimately, so if I estimate sigmas based on those questions 1A through D, you know, how does that compare to 0.36 from the historical biomass approach by Ralston et al.? Let's start with a pictorial representation of the experimental design. Let's say I want to start projecting from 1998. What I would do is then I'd look at my stock of interest. Let's pretend this rockfish had a benchmark assessment in 2009, another benchmark in 2013, and a third benchmark in 2015. I would pull the report files from those assessments, and then I would go in there and grab all of the quantities I'd need to do a population dynamics model, all relative to the year 1998 or 1997 to project onto 1998. All right, and so that would give me the quantities I would need to calculate the exploitable biomass. I could apply the FMSY proxy that was needed for that fish, and then project any number of years forward based on the inputs from these assessments. And then we'll get different answers depending on which assessment gave us the quantities needed to calculate an OFL. We can quantify that variation to calculate a sigma for the OFL distribution. And there's a couple of places, a couple of levers, if you will, where we can manipulate this experiment. For example, in the population dynamics model, we can add stochastic recruitment. We can use different start years. In fact, a lot of them that you'll see today are 1998, 2003, and 2008, I believe. So we can change from where temporally in their report file to grab information. So we can capture how the age structured components vary over time in that assessment structure. You can also manipulate the number of years to project, and we'll see 25-year projections to start with, and you might guess that the tactical configuration of this method is not going to use that many years of projections. And then for the calculating sigma, there's actually 9 different sigmas that we calculate. It just depends on which one of the subscripts that you care about or dimensions that we're looking at, like stock specific, or we can have them start year specific. Stochastic is another dimension, for example. And so let's tie this, seen the experimental design sketch, let's tie it back into that uncertainty we talked about before. So when we're talking about caveats for the historical biomass, we talked about how exploitable biomass, the projections would give us more access to quantifying uncertainty in exploitable biomass. So for example, I grabbed selectivity by fleet from the stock assessments, the weight retained by age and fleet, relative exploitation rate by fleet to help. Those are some of the sources of uncertainty associated with exploitable biomass. When it came to FMSY, we applied the assumed MSY proxies of 30, 45%, and 50%. Someone's happy to correct me. It's been a minute since I've thought about this, but pretty sure if 50% is what rockfish were using, 45% is roundfish like sablefish, and 30% at the time was for flatfishes in the groundfish complex. And then the other axis of uncertainty for stock abundance and age structure, some of the experimental design components that address this are the multiple start years, being able to have multiple projection period lengths, and adding in that stochastic recruitment. So let's look at some of these projections. I'll orient you to the plots and then add a couple more rows of plots. So here we have year on the x-axis and spawning biomass in metric tons on the y-axis. Each column is from a different start year. So the projections here, these are deterministic projections of spawning biomass based on this method for 25 years. This is from 1998, meaning I dug into a report file, grabbed the quantities I needed to calculate the OFL relative to 1998 in that the time series available in the assessment. This middle one is doing the exact same thing, but moving forward in time, grabbing a snapshot at 2003 and projecting from that snapshot, and again for 2008. And then the colors here are just for the, I think this is for Boccaccio rockfish. So the oldest usable assessment I could use with this method is in yellow. The one that came after is blue, solid, and then the most recent are green dots. So you can see that they're a little bit different. What's going to appear on the screen next are two more rows, and they're both stochastic recruitment. We decided to look at stochastic recruitment in two ways. I have those as auxiliary slides if you're really interested, but I'm just, for the sake of today's talk, they didn't have qualitatively different answers, so they're going to look very similar to you. So let's go ahead and add some more. Same plot, just expanded. We've got the deterministic up here and the two flavors of stochasticity here, and you can see that using the projection-based approach for 25 years, we are getting variation in what we think the spawning biomass would be based on the stock or the model structure from 3 stock assessments. Let's look at OFLs because we haven't looked at any OFL time series yet in this talk. Orient to the plot, we've got years again on the y-axis, and what I've actually done here to show you the sheer difference in scale is I imagine this row, I've just combined them, I've smushed them into one plot. So you could just see, you know, if this was 1998, how different in scale my projections of overfishing limit are from the next 2 assessments. All right, and so this is for deterministic. I'm going to expand this to show you the stochastic work. They generally have that same trend where one assessment had higher scale projections and the other 2 were lower, right? And so that's what the projections ended up looking like. I want to take a pause and talk about the assessments I was actually able to use. Relative to the historical biomass approach, we could only use a subset of stocks, 7 to be exact. Here's their common names for the fish nerds in the audience. Why only the 7 when there were a lot more? I think at table 1 in the paper will have all of the ones that Ralston used. But we wanted a benchmark assessments only because that's where benchmark assessments are where the terms of reference allow you to actually make changes to the model structure. And we didn't want sort of like false replicates of using updates because their structure shouldn't be changing in the model too much. We needed 2 or more assessments conducted for that single stock. So I needed Boccaccio rockfish to have 2 or more assessments done in some version of SS 3.24 at that time or whatever, or later. And that was because some of the quantities I needed to calculate the biomass and then the OFL from that biomass were not easily available from older than 2009 assessments for US West Coast. And then, unfortunately, for some of the coastal pelagic species, I was not able to use them because they had a seasonal model configuration, and we weren't really sure the best way to summarize across those seasons to make sure we're comparing apples to apples with some of the other, or some of the groundfish species, for example. All right, so those are some things to think about as restrictions on this method that can be used. And hopefully, in theory, as you all do more assessments, we would be able to increase the number of stocks represented on this list. I'm going to give you a very simple slide on how sigma is estimated. I say it's just a run-of-the-mill variance with some sprinkles of species, projection year, start year, and stochastic dimensions. I'm just going to show you 2 of the equations in table 1, which I think has way too many, but they all have this general form, very similar to the Ralston et al. Method, where there's some mean from our projections. We have the actual, so the square deviations based on the mean and the observed simulation projections, and the differences between all the sigmas, which I'll show you in a picture form very soon, are just the number of summations you have. So which dimensions are we going to sum over? Let's look at that picture like I promised. Here are the 9. We're just summing over whether it be the start year, a species or stock, a projection year, and you're just collapsing over those. We can broadly look at among assessment variation and within assessment variation. I'll give you the spoiler for within assessment variation. For all the assessments that we looked at in this study, there was hardly any within assessment variation. They were very tight projections when you looked at them with all these different dimensions. And I'll just say the one to watch is the sigma bar here, where we just pull over start years, the stocks, the 7 stocks, and the projection year scheme that we have going on. Let's look at some sigma over time. Apologies to the handout, folks. I definitely left extraneous notes here that you don't need to pay attention to. So let's look at a different plot. Now we have projection year on the x-axis. So this is a 25-year projection period. On the y-axis, we have the actual sigma values themselves. This would be, I think, sigma YSP for those that love notation. We have the different start years as the different colors. And then if you pull across all 3 of these years, you get that solid black line. And then the column on the left is projections of spawning biomass and projections of OFL on the right. And what I'm going to show you now is adding in the stochastic considerations that we had. And we can see that looking at variation based on start year for the projections can yield different results. And this is awesome. We want to see this because we can start to characterize or quantify the uncertainty in that and add that into that P-star harvest control rule. So let's look at a comparison of the historical biomass versus projection base. So 25 years of projections with 3 start years, 1998, 2003, 2008, it's every 5 years. If we use just spawning biomass, here's that row of data with the 0.4, stochastic and deterministic will have a little bit of difference. And then the OFL itself, here's that 0.36 value from the historical biomass approach. I'd like to show this to you in picture form in addition to just table form. Not everyone's a big fan of the table. Here's what the historical biomass approach looks like in terms of the shape of the OFL distribution because of the sigmas. Remember, this is the 0.36, the doubling, and the doubling again. But the projections, we actually go from 0.36 for category 1 to 0.5 for category 1. So we're getting a little bit more stout, and then you're doubling of that and doubling again. And so in theory, we're getting a bigger buffer between the OFL and the ABC to account for that scientific uncertainty we've quantified using this method. I'd like to use the rest of my time to briefly describe what tweaks I made to some of these levers to address questions from the SSC as they reviewed this. I think I presented it 3 times, once in a proposal phase and a kind of like, are you even interested in this type of analysis kind of deal, invite back to see the 25-year academic exercise. And that's when they gave me more specific questions so I can produce results that are in like units that are actually applicable to the kind of work that's done at the council, and then a final review. Maybe a week or so before my master's defense. Some say to this day that the SSC was more difficult than my master's defense, but it's only alleged now. Okay, so what did the Pacific Council SSC want to see? So here's that sigma plot again. But now what's different is the projection years scale here. Instead of 25 years, we're having from 1994 to I think 2008. I'll have to double check that. But basically, instead of grabbing, say, 1993 and projecting for 25 years, they wanted me to grab 1993 from the report file, project it 1 year. Tactical management decisions happen on short time scales. So project on a short time scale is what I was hearing. So each one of these dots is a 1-year projection with some uncertainty on it from replicates for Boccaccio rockfish. So each one of these dots is a different snapshot from the assessment models and giving us some uncertainty there. I'm going to expand this plot now for the other 6 stocks and the final and then a plot that pulls across the 7 stocks total. Let's go. Here's Boccaccio. I want to, really important, the top row has a different y-axis because there's these estimates had lots of wiggling, lots of variation, and this bottom row it has 0.6, which I think is pretty interesting. Just another example of why picking multiple start years for projections can be useful for quantifying uncertainty in our age structure and model structure assumptions. Let's zoom in to this final panel. This pools across all these stocks, the other 7 stocks, and you see you've got a little bit more stability, and this blue dashed line is that sigma hat I said folks were caring about for the harvest control rule itself. All right, it's pretty interesting to see how the stock assessment structure and just other kind of considerations can have quite a bit of variation, or maybe not so much, across the different life histories we see represented by these stocks. Okay, coming to the end. Here's some more questions in SSC style. What happens if you add new benchmark stock assessments and rerun the Ralston or the historical biomass approach, right? Those only had, I think, stock assessments up through 2009. Paper was published in 2011. People have been doing stock assessments since then. What happens? Well, their original estimate was that 0.36. If I added in all the benchmark assessments that have been done since 2009 for the same stocks used in the historical biomass approach, and we got 0.4 as sigma for that OFL distribution. But another question, my projection-based approach that we did only uses 7 stocks. What if you ran the biomass, the Ralston work, with just the same 7 stocks instead of the whole suite available? What does sigma look like then? We get a 0.35. We're using just the 7 stocks. So these were good enough sensitivities for the SSC to continue giving the projection-based approach the light of day at their council. So here is the table form again. You've seen these quantities here. For the tactical approach of 94 to 2008, we see new values based on deterministic and stochastic. And I think, here we go, yep. Ultimately, what the council selected was, or not the council, sorry for that, the SSC selected was the 0.452, which they rounded up to 0.5 as the quantity they wanted to adopt and update the 0.364. So let's look at it in pictures again. So before I did my master's and presented this approach to the SSC, this is how category 1, 2, and 3 OFL distributions were defined for the P-star harvest control rule to set the ABCs, or these red lines. With the projections-based approach, we see an update of that category 1 to 0.5, so more stout, bigger buffer, and then the doubling for category 2 and 3 follows through. I should note, I don't have any slides on this, but I should note that I showed you that picture of Chantel and I being reviewed at the same time. Chantel was presenting work she did with Owen Hamel on what I think is unofficially called the accelerator, where that sigma value of 0.5 is actually increased as that assessment ages up to 10 years. So if it takes 10 years to do another benchmark assessment, or maybe update, don't quote me on that, do another assessment, then that the model uncertainty with that dormant assessment should increase over time. That's a cool thing to look forward to reading about. And yeah, I just want to sort of wrap up and say that the projection-based approach captures more sources of uncertainty than the historical biomass approach. And the SSC decided to adopt that for US West Coast groundfish and coastal pelagic species. I want to make a note that accounting for uncertainty in the recruitment, the two different methods we used, does make the calculations more complete, and we did use the stochastic sigma, but it was noticed that it's not qualitatively changing the results very much. Some caveats, of course, not models are simplifications of reality, and they're going to have problems. And unfortunately, the projection-based approach does not overcome the very common problem that's assuming stationary parameters are non-stationary is problematic. And we still see the same culprits within the assessments that contribute to this overall uncertainty. That would be your growth, natural mortality, and productivity parameters. So, that's still very much still there. Some of the caveats I will roll into a optimistic future work slide here. You can expand this approach to include assessments with more complex structures, like those seasonal models for sardines that I mentioned, and spatial models. Pick your assessment model flavor. Several older assessments needed to be excluded because they were missing some key outputs needed to project a biomass and/or the overfishing limit itself. And so as folks continue to do more assessments, we can start adding more stocks into the pooled sigma that's calculated. And folks at the council, and by folks I mean like members, stakeholders that are a part of the other committees that then communicate to the council themselves their concerns, are interested in exploring stock-specific sigmas instead of pooling across all 7 stocks, for example. And I think from like the analyst's point of view, as more assessments become or get added, I imagine that this could become a reality. I cannot make a prediction about if that actually makes people happier or not, but fortunately that's not, I think, something that I have to worry about. My overall recommendation, Andre and I's overall recommendation, is if you're going to use a projection-based approach to define the sigma used in your OFL distribution, we recommend that your assessment, whether you do it in stock synthesis 3 or not, provides projections from various start values, because this allows those projections to be, or projections based on multiple assessments, to be included in the calculation of that among assessment variation and capturing just yet another source of uncertainty. And so to show the same recommendation in picture form, remember how all these different start years for the tactical version of this project varied widely for some stocks, right? So if you are going to use the projection-based approach, please consider using as many start years as feasible so that you can capture the variation that we see here. And with that, just a little bit over 45 minutes, I want to say thank you, and I want to acknowledge that this talk was vastly improved by the support of my Punt Lab mates, like graduate students, postdocs, and alums, and of course, Andre Punt himself. And I'll take any questions as long as time allows. I'll leave my contact information here.
Excellent. Thank you so much for your presentation, Kristin. I want to let the audience know that we have plenty of time for questions. We've got about 10 minutes. So if you have one, please type it in the questions chat box, which is part of the GoToWebinar control box on the right, and I'll read them to Kristin. And while we wait for some questions to come in, I also want to remind you that Kristin did share her slides, and they were really awesome. Thank you for putting them together. So if you're interested in having a copy, go ahead and download them before we end the program from the handouts menu in the control panel as well. And if you joined us a little bit late, this is recorded. So if you know, I have a colleague that might be interested in Kristin's work, I'm going to upload the recording to the NOAA Central Library YouTube channel. The link is in the chat if you're interested, and that will be available in just about 2 or 3 hours. So.
And I don't know if this is the right forum to discuss it, Lisa, but I would love to hear from folks that work in other council regions to hear about, do you use the P-STAR approach? How do you quantify the uncertainty in it? I have read one of the national SSC reports from 2015, and that looked, where I got to see a lot about how you are all using ABC control rules to go from an OFL to an ABC. But there's a lot of variety in how folks do that, and it's not necessarily with the P-STAR. And I'd love to, we're well past 2015, so shoot me an email if you're also a nerd about this stuff and you want to have e-coffee about it, or if time allows, I'd love to hear what people are doing.
No, it's perfect. I'm happy that you're asking questions. Again, all of Kristin's contact information is right there on the screen, as well as the handout that she provided. So if this is not a good time for you to ask the question or to respond, you can do that personally offline. I'm going to give us another minute, Kristin. I'm so glad that the leaf blowers did not interrupt. All right. Well, I'm going to end the program here then, since we don't have any live questions today. Again, please feel free to contact Kristin offline. Thank you for your presentation. And also to Kristin Blackheart, thank you for organizing the National Stock Assessment Science Seminar Series. I hope you will join us at NOAA Central Library for another National Stock Assessment Science Seminar, which are hosted on the first Thursday of the month at 3 p.m. Eastern Standard Time. So see you next month, and have a great day, everybody. Bye-bye.